Hello students, welcome back to a new video of Sunanda's tutorial. Today we will discuss about the classification of Bryophyta. In my previous videos I have already discussed about the salient features of bryophytes, then the life cycle, general life cycle of bryophyte. Today we will discuss only about the classification. So the word Bryophyta was first used by Brown in 1864. He included algae, fungi, lichen and moss in this group. But later on, algae, fungi and lichens were separated as Thallophyta and liver rot and moss as Bryophyta. The rank of division to Bryophyta was first given by Simper in 1879. He treated Bryophyta as a division. Later on, Eichler in 1883 recognized two subgroups of Bryophyta. One is Hepatici and one is Mossi. Since then, these two classes, one is Hepatici, one is Mossi, are separate major entities within the Bryophyta division. Then Engler in 1892 subdivided each of the following classes into three orders. So Engler divided Bryophyta into two classes. One is Hepatici, second one is Mossi. Hepatici again subdivided into three orders: Marcantiales, Jungermannials and Anthocerotales. Similarly, Mossi divided into three orders: Pagnales, Andrels and Bryals. Chopra in 1981 considered Bryophyta to be triphyletic, and the three subdivisions of Bryophyta proposed by him are Tachyon-Phytina, Hepatophytina and Moscophytina. The anomalous position of Anthocerotales as an order of the class Hepatici was pointed out by many investigators. But later on, who in 1899 gave the class status to the order Anthocerotales and he named it as Anthoceroti. So after him, the Bryophyta divided into three classes as Hepatici, Anthoceroti and Mossi. Many eminent hepaticologists liked or supported this system of classification. But Scoster in 1958 called Anthocerotes as Anthoceroti. So after him, the Bryophyta division was classified into three classes. One is Hepatici, second one is Anthoceroti and the third one is Mossi. Rothmaler in 1951 suggested the following class structure for old ones. He replaced Hepaticopsida for Hepatici. He named Anthocerotopsida for Anthoceroti and Bryopsida for Mossi. So these three names were suggested by Rothmaler: Hepaticopsida, Anthocerotopsida and Bryopsida. The new names suggested by Rothmaler have been recognized by ICBN, which is International Code of Botanical Nomenclature. Proskier in 1957 suggested the name Anthocerotopsida for Anthoceropsida. The modern bryologists thus classify Bryophyta into Hepaticopsida, Anthocerotopsida and Bryopsida like this. And many orders are placed under these three groups. So what is the basic features of Hepaticopsida, which is the first class of Bryophyta? The plant body is thalloid or foliose. Foliose means foliose-like or a leaf-like structure. Rhizoids are present, that means root-like structures are present but without septa, means cross wall. There is no cross wall between them. They are simple or tuberculated. Simple or tuberculated. Tuberculated means dented. Ventral scales are also present. Cells have chloroplast but without pyrenoids. Green cells contain simple or compound oil bodies as reserve food material. Sex organs develop from superficial cells on the dorsal side of the thallus, except when they are terminal in position. Sporophyte, simple and differentiated into food, seta and capsule. Sporogenous cell develop from endothecium. Columella is absent. Generally in higher bryophyte, one sterile part is present in the sporogonium, which is columella, that is totally absent in case of Hepaticopsida. But illators are present. Illators helps in the spore dispersal. I have already discussed that spores are formed in sporophytic phase inside the sporogonium. Inside that sporogonium, many illators are present, and that illators are help in the spore dispersal. So it is present. Sporophyte completely dependent on the gametophyte. Sorry, it is the gametophyte. Sporophyte is completely dependent on the gametophyte for their nutrition. Dehiscence of sporogonium is irregular or regular. Then Anthoceroptocida, it is the second class under the Bryophyta division. Plant body is thalloid or dorsiventrally flattened. Dorsiventrally means it have a dorsal side and ventral side. Rhizoids are simple without septa, that means cross wall are absent. Ventral scales are absent. Generally in case of Hepaticopsida, these scales are present, but in Anthoceroptocida, the scales are totally absent. No tissue differentiation in thallus. Cells bear large chloroplast with conspicuous, that means large pyrenoids. Sex organs embedded in thallus tissue. Antheridia, that is male sex organ, develop from hypodermal cell of the thallus on the dorsal side, located in antheridial chamber, single or in groups. Archegonia, that is female sex organ, are sunken on the dorsal side of the thallus, develop from superficial cell. Sporogonium, elongated and cylindrical, arise from the dorsal side of the thallus. Sporogonium has a foot, a meristematic region and capsule, that means a true seta is totally absent. The meristematic region is intercalary in position, and due to sporogonium continues its growth throughout the growing season, they helps in the growth of the sporogonium. Capsule wall has chlorophyll and stomata. Capsule wall. Capsule wall means the sporophytic generation consists of food, seta and capsule, and capsule wall of the outer layer of the capsule have chloroplast and stomata. Columella is sterile part present in the inside the capsule, which is present, and pseudo illators are present. They are not true illators. Illators like substance are present inside the capsule, that is pseudo illators. This is the characteristic feature of this Anthoceroptocida. Sporogenous tissue develops from amphithecium, but in case of Hepaticopsida, it is developed from endothecium. Then Bryopsida, the primary gametophyte consists of postrate filamentous or thalloid protonema and erect gametophore. The adult gametophyte consists of stem, spirally arranged leaf, and sex organs are apical in position. They have not true stem, but they have stem-like and leaf-like organs. Rhizoids multicellular and with oblique septa. In case of Anthoceroptocida and Hepaticopsida, these cells are not, these rhizoids have no septa between them. Then sex organs develop from superficial cells with two cutting faces. Sporophyte differentiated into food, seta and capsule. Capsule wall has stomata. Sporogenous mass develop mostly from the outer layer of endothecium, which is in addition forms columella. Illators are totally absent. Peristome is present in majority of mosses. A peristome layer is present in the capsule. Then this is the recent classification of bryophyte, where the all bryophyte grouped under three divisions. One is Marcansiophyta, one is Bryophyta, the third one is Anthoceuroptophyta. Marcansiophyta is known as liver rots, Bryophyta, mosses, Anthoceuroptophyta, horn rot. Again, these divisions are classified into many classes. So, it is the recent classification followed by many eminent bryologists. So, it is all about your classification of Bryophyta. In my next class, I will discuss about the economic importance of Bryophyta. So, stay tuned and enjoy my videos like this. Thank you.